Ladies and gentlemen, we're here to plan a prison break. Not for criminals, but for life-saving drugs trapped in cellular lockdown. Our mission? To bypass a security system that's been foiling drug delivery attempts for decades. Now, imagine a life-saving drug that has bypassed the defenses of the human body and made its way to a target cell through the circulation. The cell now engulfs the drug and traps it in a membrane-bound compartment called the endosome. And now a race against time begins. Can the drug escape the endosome before it's lost to degradation? Because as the endosome matures, it acidifies and fuses with digestive lysosomes, which releases enzymes to break down the drug before it can make its way into the cytoplasm and exert its therapeutic effect. In fact, only 1 in a million molecules may escape the endosome, and over 99% will remain trapped in there, unable to fulfill their final function. This is called the endosomal escape problem and is a major issue for a range of therapeutics, from classical small molecule chemotherapeutics to novel gene therapies. So scientists have thought, what if we had an agent on the inside who could help the drug escape from the endosome? So we quote from a famous review: a safe endosomal escape agent should have low toxicity, high efficiency, ease of use, modular attachment of targeting ligands, and cost-effective large-scale manufacture. Now, attempts to create such an agent have occurred over multiple decades in multiple chemical spaces, but none have managed to fill all these criteria. For example, in the 1980s, cationic lipids were developed to fuse through the endosome, but they're toxic because they integrate with the cell membrane. In the 1990s, the proton sponge effect was exploited, but it's hard to engineer because we don't understand how it works. And most recently, cell-penetrating peptides have been used to open pores in endosomes, but they're highly inefficient. And worst of all, none of these designs are modular because of size limitations. So, because there is no solution to endosomal escape, the current strategy to compensate for inefficient drug delivery is to increase patient doses, which increases the risk of off-target toxicity and increases the cost for patients in the healthcare system. And lastly, it reduces the maximum effective dosage you can give to a patient. But what if the perfect endosomal escape agent has been just waiting for us all along in a humble freshwater bacterium?
We're the REBolutionaries, and we're introducing R-bodies. R-bodies, or refractile bodies as they're called, are insoluble, enormous protein polymers found in bacteria. They are coiled in neutral pH, but extend into sharp needles in acidic pH, allowing them to burst cellular compartments. R-bodies measure up to 0.5 micron in diameter when they're coiled, and extend up to 20 microns in length. But they are inherently non-cytotoxic to human cells. They can survive harsh treatments like boiling and sterilization, which makes them extremely stable. In nature, cytotoxic bacteria use R-bodies to puncture food vacuoles and deliver toxins to enemy paramecia hosts. But we want to repurpose R-bodies to burst endosomes in human cells, as the acidic environment of the mature endosome will cause the R-body to extend and burst the endosomal membrane. If we can attach drug molecules to R-bodies via an acid-labile linker, the drugs will cleave off as the R-body extends and bursts the endosome, facilitating endosomal escape of the drug into the cytoplasm of the cell. Our system will greatly enhance drug delivery efficiency, which lowers dosage requirements and minimizes costs and off-target toxicity. It is very customizable, and it can be used in a range of applications with any combination of drugs or targeting molecules. We can use it for anti-cancer chemotherapeutics, RNA delivery, or gene therapy. Now, in order to experimentally validate our system, we managed to achieve the following things. First of all, a novel R-body purification method. Second, successful conjugation of molecules, including aldoxorubicin, a chemotherapy drug, onto our R-bodies using 4 orthogonal conjugation strategies. Characterization of our modified R-body constructs, verifying their functionality after modification. And successful endocytosis of our cargo-carrying R-bodies into mammalian cancer cell lines. But to be able to work with R-bodies, we first had to purify them to a sufficient extent, but we ran into a couple of problems. When we tried to use previous protocols from recent papers on R-bodies, we obtained this stringy, indistinguishable pellet, and this microscopy image is similarly messy. So after previous protocols failed, we took 6 months to design our own purification protocol to achieve sufficient purity for downstream applications. Here, you can see that our protocol produces a clean white pellet. And in our microscopy image, you can see homogeneous clumps of small coils, which is what we expect contracted R-bodies to look like at pH 7.5. And when we placed our samples in acidic buffer of pH 3, we saw that they extended into long, sharp needles, confirming that we had successfully purified functional R-bodies. Now, achieving optimal purity is absolutely essential for therapeutics, because therapeutics need to be manufactured free of any contaminants or unknowns in order to be safely used by patients. This was something we learned from our interview with Professor John Rasko, who's a key player in putting gene therapies into clinical use in Australia.
So we wanted to conjugate drugs or other ligands onto R-bodies by conjugating them onto the monomers, the ends of the monomers that make up the polymer. To do this, we are required to design multiple independent or orthogonal conjugation strategies that can be used simultaneously, so we can attach many different types of drugs or ligands to the R-bodies at once for a truly modular or customizable delivery system. I'll walk you through these now. But in the end, we chose 4 different methods that work orthogonally or independently. Sortase conjugation, amber stop codon suppression, 2-PCA modification, and maleimide chemistry. Sortase conjugation allows us to attach proteins or peptides onto our R-bodies. So you see the sortase enzyme up there on the left is recognizing the different peptide motifs that we've added onto the R-body monomers and onto our protein cargo. It then recognizes these motifs to join them together. Protein therapeutics generally have a lot of trouble penetrating cell membranes. So if we combine them with our R-body idea, this could really make this new field of therapeutics viable. Next, we have amber stop codon suppression. You see up there, there's a tRNA that recognizes the UAG stop codon that we've added onto the gene for R-bodies. This tRNA adds any unnatural or non-canonical amino acid onto the R-body during protein synthesis. And by exploiting the chemical properties of the unnatural amino acid that we choose, we can then attach ligands or other small molecules onto the R-bodies as we choose, for example, through click chemistry. This is highly scalable, as the conjugation step is only a 1-step process, and allows for even more modularity, as we can attach any unnatural amino acid to our R-bodies. Next, we have 2-PCA modification. This is a purely chemical approach where a 2-PCA chemical attaches to the end of a monomer and then provides it with a chemical handle with which we can use click chemistry to attach small molecule ligands. This is great for researchers and drug developers because not needing genetic modifications allows them to iterate over conjugation designs quickly and will further enhance the research efforts to develop therapies. Finally, we have maleimide chemistry, an extremely common conjugation strategy which is already used for multiple approved therapeutics. So on the cargo, we have a maleimide group which joins to the thiol group that we've added onto the cysteine of an R-body. So this is a highly specific conjugation strategy which is important for therapeutics, and again, it is really common. So labs will generally be able to implement it quite easily. So we validated that these conjugation methods were successful with a series of fluorescent dyes. So up there on the left, you see that white pellet. It's our R-bodies washed through, where the dye is being washed through. And it's just a big insoluble white pellet. Just over to the right, we have the sortase conjugated R-bodies, where our fluorescent protein, M neon green, has been modified to attach to the R-bodies. For the rest of the conjugation strategies, we used a small molecule blue dye. and we see a lot of bright colors, which indicates that our conjugation strategies have generally been successful. On the far right, we see aldoxorubicin, which is that chemotherapeutic, which we joined using Leyline chemistry. And we get a nice bright red pellet, indicating that we can actually attach real drugs to our R-bodies. But while we had shown that the conjugation strategies were successful, we further had to show that cells would actually take up these conjugated constructs.
So, in answering the question of whether our cells were endocytosis constructs, we had a resounding yes. So what you're seeing here is mammalian cell culture, cancer cells from kidney cells, cultured with one of our constructs that is attached to M-neon green, a fluorescent protein. When we incubate these samples, what we find is that in the medium, you have a very dilute amount of our protein fluorescing. But when you look at the cells, we find that they're highly concentrated. Now, we wanted to verify if this is its background, right? How do we actually verify that these constructs have actually gone into our cells? And the way that we do this is to incubate the cells with a molecule called trypan blue. Trypan blue quenches the fluorescence of our protein in the medium, but it can't pass through cell membranes, meaning any fluorescence that we're seeing is actually coming from the molecules that have been endocytosed by these cancer cells. Now, we want to actually quantify how well these cells can uptake our conjugate. So what you're seeing here is a graph of the number of R-bodies that have been taken up by these cells. So on the x-axis is our experimental conditions. So we incubated the cells for either 24 hours or 48 hours, and we did that either in 1 in 8 dilutions or 1 in 16 dilutions. And on the y-axis, we just have the counts of these R-body constructs. So the two conclusions that we take from this graph are, one, irrespective of how long we incubate the cells with our constructs, there seems to be the same amount of endocytosis. So what that tells us is that the kinetics of endocytosis of these constructs is fast. And secondly, what we conclude from this is that the uptake of our constructs is in a dose-dependent fashion. The less dilute they are, the more gets uptaken. Now, this is exactly what we want for a drug delivery system. You want the effect of the drug to be proportional to the amount of drugs delivered. So it's rather remarkable that such a large insoluble protein can be uptaken by these cells, and it shows the promise of our system for drug delivery. In addition to that, the cargo molecule we attached was a fluorescent protein. It's on the order of 30 kilodaltons. It's a large protein. Any drug on the market is going to be at a much smaller size. So the fact we can get such a large protein into cells means that we can likely get smaller drugs into cells as well. And that's precisely what we see here. Here we have the same cell culture lines incubated with our samples that are conjugated to our doxorubicin, that cancer drug. So what we're showing here is that we can actually get a cancer drug into our cells with that drug delivery system. So in summary, we think this shows promise for using our system as a drug delivery system, and potentially with many more types of drugs.
Ultimately, our work is really only the tip of the iceberg. Our next step is to verify that R-bodies are actually bursting endosomes. We also want to verify that we can release the drug using acid-labile links or protease cleavable linkages. That way, once the endosome is burst, the drug can dissolve into the cytosol and go to its target site. As suggested by Dr. Christina Cook, we also need to investigate the cytotoxicity of R-bodies to verify that R-bodies alone are insufficient to kill cells. Now, R-bodies we saw could be taken up by cells in the tens of R-bodies, and this did not kill the cell. So there's really quite a lot of promise with our present work. Experts also suggested that our R-body system might have some immunogenicity problems in vivo. So one way we could potentially solve this is by coating our R-bodies in polyethylene glycol, these little squiggly bits that we see attached here. And this has been used in various nanotherapeutics already existing, and it simply serves to block any antibody interactions with the drug and reduce those immunogenic properties that we want to avoid. Now, let's explore the massive potential that our system has with commercialization. We've already shown that we can attach drugs with 4 separate, completely orthogonal techniques. So we can really attach many, many drugs, especially once we develop more conjugation strategies. Now, in addition, our R-body system relies on existing strategies, bacterial overexpression, small drug conjugation, Gibson assembly, PCR, and this makes it very easy to scale as well. Now, they're also incredibly stable, and so this makes them very well suited for long-term storage and transport worldwide. Ultimately, through our entrepreneurship work, we plan on forming a startup and potentially patenting our work in the future if it flourishes into what we want. Ultimately, with our relatively short work on R-bodies, we have already shown that they have the potential to REBolutionize drug delivery. This is because they solve the endosomal escape problem, the key rate-limiting step for the delivery of many nanotherapeutics. Ultimately, through our R-body system, we are setting drugs free to set patients free. I'd like to take this moment to thank our supervisors, Yuhang, Clement, and Connie, for all the guidance. It's really been amazing, and we're very thankful for the opportunity. Also, our university, University of Sydney, the Department of Structural Biology, and any other institutions that have helped us along the way. It's very much appreciated, and we're very grateful. Thank you.
Woo! Thank you guys so much. Well done.